Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. In today's histology class of the endocrine system, we are going to discuss the differentiate between various histological zones of the adrenal gland. So adrenal glands are the triangular shaped glands. They are present at the superior pole of both the kidneys and are embedded in the perirenal fat. They are covered by the thick capsule, and this capsule sends in septa and divide the parenchyma. So adrenal glands secrete both the steroid hormones and the catecholamines. Both the zones, the steroid hormone and the catecholamines, they are secreted from different zones of the adrenal glands, and embryologically also having the different origin. The cortex is derived from the mesenchymal mesoderm, and the adrenal medulla, which secretes catecholamine, are derived from the neural crest cells. So the adrenal glands are covered by the thick connective tissue capsule, which sends trabeculae into the parenchyma, and these trabeculae carrying the blood vessels, nerves to the substance of the gland. Secretory parenchyma consists of cortex and medulla. The cortex is the steroid secreting portion, and it lies beneath the capsule and constitutes nearly 90% of the gland by weight. This one is the cortex, and this whole area is the cortex, and medulla is the catecholamine secreting portion. It lies deep to cortex and forms the center of the gland. So adrenal cortex then further divided into three zones: zona glomerulosa, the zona fasciculator, and zona reticularis. So main hormone which is secreted by zona glomerulosa is aldosterone, that is a mineralocorticoid, and main function is to maintain the blood electrolyte balance. Its main control is angiotensin 2. Zona fasciculator secretes mainly cortisol, which is glucocorticoid, and its main function to include regulate the glucose and the fatty acid metabolisms and response to the stress. Main control is the pituitary adrenocorticotropic hormone. Zona reticularis secretes cortisol and androgens, and function and control of the zona reticularis are same to that of the zona fasciculator. So this is the diagram of the adrenal cortex. This is the outer area is the capsule. Then next to the capsule is the zona glomerulosa, which mainly secretes mineralocorticoid, that is aldosterone. Next to the zona glomerulosa is the zona fasciculator, that secretes glucocorticoids, and then zona reticularis, which secretes androgen, that is dehydroepiandrosterone. So a little bit about its blood supply, which is also very important. The adrenal gland or suprarenal gland is supplied by the three sets of arteries, that is superior, middle, and inferior suprarenal arteries. These arteries, before entering into the capsule, divide into smaller branches and forms the capsular arteries. These capsular arteries then enter into the cortex and divides into the fenestrated sinusoidal capillaries. These fenestrated sinusoidal capillaries then travel through the zona fasciculator and zona reticularis and enter into the medullary capillary sinusoids. Another blood vessel which is called the medullary arteriole. This directly starts from the branch of the capsular artery. It traverses whole of the cortex, enters into the medulla, where it divides into the medullary arteriole. So medulla of the adrenal gland has dual blood supply. It receives venous blood through the arteries which are coming through zona fasciculator and reticularis, and it receives arterial blood through the medullary arteriole. Another very important feature about the medullary vein of the adrenal gland is that the tunica media contains conspicuous longitudinally arranged bundles of smooth muscle cells. So normally we have studied that the tunica media is formed by the circularly arranged smooth muscle cells, but in case of the medullary vein, this is an important obvious feature that the tunica media is formed by the longitudinally arranged smooth muscle cell, and it is also very important MCQ. First we will study the zona glomerulosa. The cells in zona glomerulosa are closely packed and arranged as ovoid clusters and curved columns. Also to remember that in humans adrenal glands, some areas are missing the zona glomerulosa, and direct beneath the cortex there is zona fasciculator. So the cells of zona glomerulosa are small, either columnar or pyramidal shape with the spherical nucleus. This is the transmission electron micrographic picture of the cells of the zona glomerulosa. Here you can see nucleus which is present with the peripheral heterochromatin present, smooth endoplasmic reticulum which is abundant, also mitochondria with shelf-like crystals, and these are the large mitochondria. In addition to smooth endoplasmic reticulum, well-developed Golgi apparatus is also present, and these two features shows that this is a steroid secreting cell. Here you can see mitochondria as well. Free ribosomes can also be seen, and sparse lipid droplets may be present in it. So zona glomerulosa cells, smaller zone, nearly 5 to 10% by weight of the adrenal cortex, pyramidal or columnar shape, typical characteristics of the steroid secreting cells. This is H&E stain of the adrenal cortex. Here you can see the capsule which is dense irregular connective tissue. Just beneath capsule, this zone is zona glomerulosa, and here the cells are arranged either in curved columns or ovoid masses. Next is the zona fasciculator. Here you can see curved columns or curved masses of the zona glomerulosa which are present just beneath the capsule. So this is the diagram of the H&E staining of the zona fasciculator. Here you can see the cells are either two cell thick, or at some places these columns are one cell thick, and in between these columns you can see the fenestrated capillaries which are present over here. The cells are vacuolated, and these vacuolated cells are because of the lipid droplets which are washed during the process of the slide formation. And this zona fasciculator totally constitute nearly 80% of the volume of the adrenal cortex. This is the electron micrographic picture of the cells of the zona fasciculator. Here you can see the nucleus of the zona fasciculator. Large number of the mitochondria are present over here, and also the smooth endoplasmic reticulum, and in addition large number of the lipid droplets are also seen. This inset show large number of the smooth endoplasmic reticulum and Golgi apparatus which is well developed in this case. This is another beautiful diagram, this transmission electron micrography, the two cells of the zona fasciculator. Here is the nucleus of one cell, lipid droplet, and this is another cell, and these are the lipid droplets which are present. In addition, you can also see the mitochondria over here which are larger mitochondria, and then you can see the endothelium which is present. This is fenestrated endothelium, and this is the nucleus of endothelium, and here you can see the capillary lumen. Next is zona reticularis. This is a smaller, deeply staining area, nearly constitutes about the 7% of the cortical volume. The cells are arranged as anastomosing cards separated by fenestrated capillaries, again having well developed smooth endoplasmic reticulum, numerous mitochondria with tubular crystal, and little rough endoplasmic reticulum. So this is the diagram. Here you can see zona fasciculator, and this is the zona reticularis, and then the medulla. So here you can see deeply staining cells of the zona reticularis and columns and cards of the zona fasciculator. Another stain at low magnification, this area is the capsule, then is the zona glomerulosa. Here you can see the curved columns and cards of the zona glomerulosa, then zona fasciculator. This area is zona reticularis, and here the medulla starts. In this diagram, the higher magnification, this area is zona reticularis. Here the zona fasciculator is ending. Here you can see the adrenal medulla, and this one is large medullary vein which is present. Another diagram at lower magnification, you can see the capsule, the perirenal fat, then zona glomerulosa, fasciculator, reticularis, adrenal medulla. In this higher magnification, this is the zona fasciculator, and zona reticularis is present, and beneath zona reticularis there will be the medulla. Another diagram showing you this is the area of the zona fasciculator, zona reticularis and the adrenal medulla, and this area shows the medullary vein, and this area is shown a higher magnification from the area of the medullary vein, where you can see longitudinal arranged smooth muscle cells of the tunica media, which is a very important point from MCQs as well. So this is electron micrographic picture of the cells of the adrenal medulla. This is the cell which secretes norepinephrine, and this one is the cell which secretes the epinephrine. So here you can see the difference, that both of these cells contain dense core granules which are present in both the type of cells. These granules contains the hormone, the norepinephrine or epinephrine, which is secreted, but the granules in the cells of the which secretes norepinephrine are more numerous and larger and more dense. So this is a feature which differentiate the cell which secrete norepinephrine from the cells which secrete epinephrine in electron micrographic picture. So here I complete my today's class and also the endocrine system. Here are the references: the basic histology by Laikus and Siddiq, the histology text and atlas by John Quera, and histology text and atlas by H. Ross. Thank you very much for your attention, dear students. Inshallah, we will meet in the next class with new system. Till then, Allah Hafiz.